Well, thanks, Munir. So to follow in the footsteps of the other speakers, I will tell you briefly about me. I was thinking about how to actually start this off. First of all, as you see, I am on the board of NHS England. I joined with Noel Gordon, interestingly, Noel Gordon and David Roberts from Nationwide. I chair a company, and I'm going to talk about that significantly in a few minutes. I'm a consultant. I've been a consultant since 1984. I joined the medical profession officially in 1976. I was a medical student from 1970, and I used to hang out with our local GP and drive him around because he was an alcoholic. And so from the very earliest days of my medical career, I was sticking needles in people and prescribing. But in fact, before all of that, I was an entrepreneur. One of the products of my entrepreneurship actually is the Centre for Life. I had the idea for that while taking my daughter for an interview in Cambridge in 1994, and there was a call for Millennium Landmark projects, and I thought, genome, we'll build a gene dome. So in fact, my daughter insisted it was her idea, but I'm telling the story. We basically built that into what became the Centre for Life, which is opened by the Queen in 2000 and has Watson and Crick, or the late Francis Crick, as its patrons. What I wanted to really think about was actually what it is to be an entrepreneur, because I was an entrepreneur before I even became a medical student. This is my dad, who grew up on a hill farm in southwest Durham and was forced to leave school at 14 because he couldn't speak with a posh accent, and did his apprenticeship as a joiner and invented ice cream vans. And the very first ice cream vans, the Mr. Wheatley vans you see all over the country, he was one of the first guys to actually have the idea of putting asbestos bodies on Ford trucks. And so when I was 10, I was very proud to be the son of the local businessman. He went bankrupt when I was 11 because they changed the tax on ice cream, and for 6 months no one bought any ice cream vans, and his cash flow dried up, and perfectly good business was wiped out. He actually came back 6 years later, same story about wives. He told my mother when I was doing my O-levels that he was giving up his perfectly good managerial job to set up another company selling pit heaps. And you can see my mother's face. You can imagine that sort of face you had from before. And so while studying for my O-levels, I was picking bricks out of the back trucks because we were selling pit heaps to the power stations. Pit heaps were actually most from the 19th century. They were full of coal. And no one realised they were actually, you could burn the things. And so we then started selling railway lines, which the Beeching had closed down. They were made of coke, made steel. And we sold that to Poland. So when I was 16, I was in the back of a lorry. When I was 17, I actually drove a Mercedes to our grammar school and parked it next to the headmaster. My dad paid the fortune to get me insured so I could do that. So I actually was the son of an entrepreneur, but actually he then discovered that while we were selling coal, I then discovered I could actually use the open cast we had. I started selling coal door to door, and I discovered I was undercutting him. So I actually bought my own car with that. And I also met my wife, who's now 45 years. But actually the important thing that happened when I was 17 was I got taken to a lecture on genetics by a guy from Oxford. I never actually found out his name, but he explained how DNA worked. And on the way back to school, our biology teacher asked me to explain it to him because he hadn't understood it. And that was the first moment in my life I had one over on a grammar school teacher. So it stuck in my head. When I went to medical school, I decided to do a degree in genetics in 1972. And in 74 said I'd be the first clinical geneticist in the Northeast, and I was. So in fact, my entrepreneurship really predated my medical life. I was going to tell you another story. I'll very briefly tell you. When I was 16, actually, I was sent on a school cruise to Russia. No one else seemed to have the idea they wouldn't sell cigarettes on the ship, so I just bought cigarettes and sold them to my friends and made a lot of money, but I feel bad about that. But I did come back with more money than I went with. Entrepreneurship is not just about making money. It's about having ideas, seeing a gap in the market, filling it, and being enthused to actually drive that forward. But in fact, I think George W. Bush, when he was having a go at the French, said they haven't even got a word for entrepreneurship, which you have to think about for a moment, because this is actually a French word. You might wonder why we use a French word. We're supposed to be the business country, and in fact, if you were here 150 years ago, all the entrepreneurs were called undertakers. And so, in fact, an undertaker is one who undertakes. And the funeral director stole the name, so they switched to the modern French word entrepreneur because they didn't want to be called undertakers and get mixed up with killing people, which was what reminded me about selling cigarettes when I was 16. Anyway, very briefly, I set up a company when I started running the genetics service because my predecessor had been doing paternity testing for the courts. And our department did it. There was no insurance, there was no proper management of the account. So I asked the university, can we not make this into a company? And they said, oh, we don't do companies, we're a charity. You have it. So with a couple of my colleagues, we set up Northgene and basically funded the department Christmas do out of it for several years. It's still going strong. It's now part of Quantum DX. It actually hit the headlines last year when I was asked by the Archbishop of Canterbury to work out who his dad was. So we actually got 6 pages of advertising in the Sunday Telegraph out of that, which was rather good. And you probably all heard it wasn't Mr. Welby after all. Very good story. I then helped set up the Genetic Knowledge Park in the Northeast at the Life Centre, and I was then very involved in getting molecular diagnostics for syndromes, and I suggested in 2004 the radical idea, instead of trying to work out which gene to test, why don't we just test them all? And so we set up a spin-out company which is owned by the hospital and the university called NewGene, which is still tootling along, going strong, selling panel testing and using next-generation sequencing. So these were basically companies that I didn't seek to make any money out of. It was really just the idea of doing something different that was exciting to me. 10 years ago, I was asked to defend DNA. In fact, Lister beat me. I came third in the BMJ competition, with DNA as the greatest discovery in medicine, after clean water and vaccines, I think. So it was pretty respectable to come third. But that's Jim Watson, our patron. I was actually in Paris with him and discovered he'd never had a selfie, so I thought I had to have a selfie with Jim Watson. I called it a Nelfie because he got a Nobel Prize. But of course, it was Jim's whole genome being sequenced in 2008 that radically changed our worlds, and that you've already heard from Fiona, this deluge of data and the issues that came from that. Last year, I was at the Expo with Sean Ellard, who led the molecular team that did all the monogenic genetic research. And just to illustrate the problem of dispersal of ideas or diffusion of ideas, Sean this is from several decades ago now. This young man is now in his 30s, and he's the chief vet in the Southwest. But at 16, he was being virtually killed with insulin because he didn't have insulin-dependent diabetes, but was being treated with insulin. And Sean demonstrated that he had a mutation in a specific gene which gave him maturity-onset diabetes of the young. So he needed a very cheap oral drug and not insulin. And she took him off it, or they took him off insulin, put him on the oral drug, and he succeeded. And if you go around Britain, there are about 4,000 people have been diagnosed with this out of the 40,000 that we think are out there. If you live in the Southwest, you might well get that diagnosis. But this is the distribution of diagnosis of that condition around Britain. And this is not because there's something special about people from Cornwall. It's because the molecular lab that delivers the service sits in Cornwall. So we aren't good at diffusing what are self-evidently good ideas. And you've already heard about Genomics England, and I was the chair of the innovation panel under John Bell and the Human Genomics Strategy Group, which basically was the underpinning of the idea in 2012 to launch the Genomics England strategy. And as I'm sure you all know, about $500 million's gone into that, and I sit on their advisory team. And that's obviously a major, in terms of the theme of today, this is the big influence on our lives. And you've already heard about Congenica as one of the obvious spin-outs from that exercise. But I actually got into this genomic application in a rather interesting way. About 10 years ago, I met a young man called John O'Halloran who had been working at Harvard, and he worked next door to Charlie Lieber, who just started describing nanowires. And he had the idea that John O came back to England and said, why can't we actually use nanowires to do DNA diagnostics? We could stick the DNA molecule to a nanowire. When it meets its partner, the wire rings, you get a shifted electric impulse, and we could detect its presence. So to cut a long story short, him and Elaine Warburton mortgaged their houses, and I ended up adopting them into the Centre for Life, and we set up a company called Quantum DX. And while we're signing the Genomics England contract at number 10, I took the opportunity, never miss a good opportunity for a photograph, so I caught George Freeman, the then Minister for Life Sciences, and showed him what we'd managed to create by 2014, which was a disposable cassette that would extract, amplify, and analyze DNA in under 20 minutes. The only problem was when we showed that to Sonia, it was going to cost about 100 quid to make one, which was actually no use at all, so we went back to the drawing board and started again. Meanwhile, we were working out what we were going to do with this gadget once we'd invented it. And the atrial fibrillation challenge was already well out there, and the idea that there were 1.4 million people in this country with an irregular heartbeat. If you don't put them on anticoagulants, they have an extremely high risk of having thrombotic strokes as clots form in their heart. So an obvious target population, because they're treated with a drug called warfarin, and warfarin has huge side effects. And one of the reasons it has side effects is because some of us are very sensitive to it. And so the idea was that we would actually develop a point-of-care genetic test. Now, if you take too much warfarin, you bleed and it can kill you. And so we set off towards personalized warfarin use, while another group were working Big Pharma towards the direct-acting oral anticoagulants. So what I called the high road, certainly in terms of cost. Now, I was oblivious to that, I have to say, when we were discussing the warfarin idea, because I was influenced by Anne Daly, who's sitting at the back of the room, who wrote this paper in 1999, which is a citation classic, which identified the gene, in fact, one of two genes that identify which members of the public will be affected by warfarin. And so we have two very common variants in our population which make people very sensitive, and a third variant in a gene called VKRC1. And the way this works is this: you need clotting factors activating, you activate them with vitamin K. Vitamin K is activated by the product of a gene called VKRC1. There is a common variant in that gene. If you have two copies of it, then your VKRC1 doesn't work very well, so you're not very good at activating your clotting factors. Warfarin blocks the product of that gene. And S-warfarin. S-warfarin is broken down by CYP2C9. If you do that well, that's fine, but a lot of us don't do it well, so the S-warfarin builds up. If you have the combination of an underactive gene underneath it and a build-up of warfarin in the system, you get a massive reduction in your clotting factors and you bleed to death. And around about 1 in 30 people in our population have all three variants and are very sensitive to warfarin. So back in 2003, we wrote the white paper and said, good example, here's a woman of 58, she gets put on warfarin, misses her appointments, nearly bleeds to death. By 10 years from now, in 2013, the GP will have a device that will rapidly diagnose the sensitivity and put them on the right dose. So it never happened. We could have done that then. We had all the technology. We just didn't have the gumption to get it rolling, because we couldn't deliver pharmacogenetics on the point of care fast enough. So this seemed like an ideal target for our little company. So we got a grant from the Academic Health Science Network. There's Anne in the middle, and Farhat Kamali beside her, who's another expert in the field. And Liz Kendrick, who is a force of nature, had 2 sets of twins under 5, was a part-time GP, and was running one of my committees at the Regional Health Authority. And John Hanley, the haematologist around the service on the right, and Hirsch Sheth on the left, my brilliant postdoc, former PhD student. So we set off, we got a grant, we got money to set up the test. We set it up not on our new device, wasn't ready yet, so we set it up on a different machine using mass spec, but very fast turnaround, 2-day turnaround, we can give them the results. And then we started talking to hematologists and said, well, we need you to bring the patients in on day 4. Then we'll tell you what dose to give them. So we can't, we bring them in on day 3. So yeah, but we want them to come in on day 4, then we'll tell you what dose to start them on, instead of taking weeks to do it. Yeah, but they all come in on day 3. So we just kept going round this conversation. No one could work out how to stop bringing the patients in on day 3 and move it to day 4. As a result of that sort of problem, after a year and a half, we'd done 5 tests, despite the fact we were giving it away for free and offering complete support service. And so this was very disappointing. Now, this was despite the fact that the paper that had come out from Munir demonstrating that if you did this, it worked. Not only did people less likely to overshoot and bleed, they were also going to get into the therapeutic range more quickly. So why couldn't it work? Well, because we really didn't address the whole pathway, the whole system. You can't just hit it with a really clever gadget or a piece of software. You've got to talk to the entire system and get them working. In fact, Munir was already working on that with his colleagues in the Northwest, rolling it out through the nurses doing the ECGs, trying to get right up to the front of the game. And of course, we also had the apps, the variety of apps, which will actually tell you what dose you should be going on to. So instead of taking weeks to gently get them to the right dose, you simply work out what the right dose is, put them on it, and save several weeks of visits to the hospital. We couldn't break into our whole system, despite the fact our GPs were spending 6 times as much on the new drugs, which were now being pushed very hard because there was an enthusiasm to get new drugs onto the market and prove the NHS could do this. And what's been fascinating is that we've figured out our clinical commissioning group spending about £4 or £5 million more than it needs to, but getting them to spend a few £100 to actually switch the system around is very difficult indeed. And that was despite the fact there were problems with these new drugs. In fact, they are not necessarily quite as good as they were presented in the original papers. But importantly, they are also more of a risk for people in the older age group. Despite the fact that, despite that fact, NICE are still saying you should offer DOACs or warfarin equally. Now, in fact, NICE didn't take account of Monet's paper because they'd finished their literature search by 2013. And so they basically said that we should be offering either DOAC or warfarin. What's happened in practice is it's a lot easier to put people on a drug that doesn't need monitoring. And so they just say, just go on to DOACs. And so what we've seen is a huge surge towards the new drugs. In the last year, NHS expenditure on these new drugs went up £407 million. It was 100 million the year before. We're heading for £1 billion extra. It'll be 5% of our drug budget, twice as big as the Cancer Drug Fund. And yet, if you simply targeted your warfarin more effectively, warfarin would be just as good. And indeed, we've got pretty good evidence it might well even be better. So the device that Monea used was developed by my friends at LGC, with whom we did a lot of paternity work in the years gone by, the former laboratory of the government chemist. So we thought, let's have another go at this. So we've got 2 more grants, and Monea's got one, we've got one. And this time we're affecting the whole system. And one of the ideas I had was to take the device and give it to the haematologist. Say, just play with it. Just test the people coming through the system. And let's see the ones who've got high INRs, that you're supposed to be on 2 or 3. So these are people who are on 11.6. These are people who are liable to bleed to death. So just test them and see what happens. And what they found was that a huge number of the people who were turning up out of control had these genetic variants. So we started to win over the team in the hospital. Actually, this would be quite a neat idea. We could actually maybe get some of these dangerous people out of the clinic onto DOACs. And so we've started to get traction. We've now got it onto the ICE system, and we're about to start again rolling out genotyping to the whole community. The other piece of evidence that we've, just to push back a little bit on the DOACs, this is a paper from 2015, DOACs are excreted by the kidneys. As you get older, you're less good at excreting the drugs. So people who go into trials tend to be younger, non-comorbid patients. So the people we're treating are in their 80s, like 4% of over 80-year-olds have got atrial fibrillation. But there weren't very many people in their 80s in the original trials. And so when you actually look at the adverse events overall in the trials were less, but in the older people, they were higher. So one of the arguments that we could discuss is maybe we should have a different policy as people get frail and be thinking about using warfarin more in the older age groups. Because the other point is that actually a lot of old people don't mind going to the warfarin clinic every month or every 2 months. It's a day out. They haven't got much else to do in their 80s. So this idea that it's a terrible imposition on them to actually have their warfarin INR checked is actually not a good argument. It's very similar to when insulin, we were sold long-acting insulins because people don't like to inject themselves. We killed countless people with long-acting insulins because we had to eat all the time, otherwise you went hypo. So in the same way, this idea of getting off monitoring isn't necessarily a good idea. So this was from 2015 when we started to get some traction in the NHS and get warfarin mentioned as a possible alternative to switching to the new drugs. And this very nice paper came out in 2015 from one of the third player in the market, Edoximab, where they pointed out that if you actually genotype people, the majority of people are up in this blue group, and warfarin wins up here, whereas the DOACs are obviously more effective in that group down there. So you could actually share this market, get more people treated, because 2/3 of the people who come in with atrial fibrillation and had a stroke are not anticoagulated. So we're not getting to people. So this shouldn't be a battle, it should be a partnership. Let's use DOACs for the people who are sensitive to warfarin. Let's use warfarin for the older people and the people who can take warfarin without side effects. The other thing you can do, and we've now got a partner on the industry side, is Roche. They've developed an INR self-testing device. And basically you can test yourself and phone in your results. So you don't have to actually go and sit in the outpatients for an hour and a half. So it becomes a much more interactive process. And we've tried this in Darlington, which is not a sort of high-tech place, and even the people in their 70s and 80s like this device. And they're getting people into the therapeutic range in 80% of the time. Now, if you look at the original trials of DOACs, once warfarin performance tops 66%, warfarin becomes the better drug. Because you need people in the therapeutic range with warfarin, obviously, for it to work. So combining self-testing with genotyping, you can actually outperform the DOACs. The other piece of evidence that we're about to publish, which might well just influence the medical profession a little, is this data from IMS, where they actually went and checked to see if people took the tablets. Now, you won't be amazed to be told that if you get seen at the hospital every month, you take the tablets because you're going to get checked. That's the top line, that's warfarin. So 74% of people at the end of the year will still take the drug as prescribed. Whereas for dabigatran, the oldest of the new agents, down to 34% of people were actually taking the drug as prescribed. So clearly, if they're not taking it, it's probably not going to work. So the strong likelihood is that these drugs are not better than warfarin in real life. So you've got to look at whole pathways. This gets, I'm not going to bore you with all of this, but people are now thinking, we can actually start to build a whole pathway. We also have to take account of the fact that African Americans don't react as well on this because they have a different SNP. We know what it is now, but the FDA still hasn't identified it as a testable SNP. But ideally, what you're going to do is have an ethnic, as Fiona mentioned earlier, you want an ethnic-specific genotyping. So you actually take account of people's ethnicity when you give them their dose prediction. And this can be done. The problem is that this is a whole system re-engineering. Meanwhile, every time I go to America, I've been 3 times in the last year, every time I switch on the television, I get an advert for a Pixaban, who happen to be, the company who make this are actually funding our project, ironically. So the point is that the biggest expenditure on drugs, of all drugs, is on this drug. And the fourth in the line is rivaroxaban, which is the next one down. So there's a massive push to get these drugs out there, and all of the guidelines are saying use DOACs first. So I'm not sure we're actually going to turn this around. I think we should turn it around. I think the NHS could turn it around and actually could save itself about £500 million without hurting anybody. Whether we will or not is another question. So we're still working on our little gadget. This is what it looks like now. And in fact, we've now got it down to a $5 production. But we're actually moving into other areas. So drug sensitivity is still going to be our demonstration, but HPV, sexually transmitted disease, drug-resistant TB, there's no question that they can be used. And also tumor markers like BRAF, which identify your response to drugs and so on. So we've sort of kept going with the device, but the device now is, this is a prototype which we demonstrated in November. So it actually took 23 minutes from the blood spot going out of the cassette to actually getting the patient's genotype on the screen. And that was the clincher. I'm not going to sit here for 23 minutes to watch that happen. It's really boring to watch because basically you hadn't to do anything. You put the sample in, go for a cup of tea, come back, you got the result. But we've now switched away from nanowires because although it was a whizzo idea, it makes fantastic PowerPoint slides. They're a bit harder to use in practice. We still have the rights to them, and we haven't got it working yet, and we will. But in the meantime, we're using a good old fluorescent biosensor, which gives us 256 tests in that one disposable cassette. And the other thing we've done is developed a way of testing TB. So this is a cyclical bit of video because if you're going to test TB, you're going to catch some tubercle bacilli. The problem is there aren't enough tubercle bacilli in the saliva. And so what we've worked with Yale is to develop a thing called DEP, which can actually pull the bacteria out of your biological sample. So we can isolate organisms just by setting the electrode to the right frequency. And so that is a completely separate pre-test technology for people with infectious diseases and bacteria. And we've caught the eye of a guy called Bill, and they have now set up to develop the HPV cassette for our device, which is obviously a huge endorsement for us. We work with Fine and Global Good and so on. So what do I learn from that? Entrepreneurship is fun. Some of us, as you've seen, 4 examples, are genetically sort of wired to do it, whether we like it or not. And we carry on regardless. And I love the NHS, and I want to be entrepreneurial in the NHS, but it is difficult. You've got to engage with the whole pathway. You've got to actually look at the entire system, not just think of a brilliant idea you can drop in. The system's current complexity, which Tony's helping to fix, is a huge deterrent. Small companies, you die if you run out of money if you can't pay the wages. It's not like you can just kick the can down the street like you would in a hospital or somebody will bail you out. Your company's dead, it's dead. Genomic medicine is focused on big data solutions, and so most of the entrepreneurial opportunities are going to be around informatics and applying that information like the way that Fiona talked about, and I'm very interested in too. But I think point-of-care diagnostics has a huge appeal because we can do things like infectious diseases out in the field. And so we're going to be piloting out in Africa under a tree with a battery-operated gadget. And that's thinking worldwide. And we just last week got 3 million from investors in Singapore who are incredibly enthusiastic about pushing this forward. But I think the small margins that you work with, in diagnostics, you don't have the billion-dollar blockbuster in these sorts of technologies. You always, even Illumina, are looking over their shoulder at BGI and all the others as the next technologies are coming down the track. So it's very unusual to make very big money. So you've got small margins. So the NHS teams we need engagement, as you've had demonstrated from Tony, that supports companies through that early phase and partners with them and puts the brand on. Because Andy said, if you're just having the brand on Congenitor is a huge asset. And that's where I'll stop. Thank you for your time.